Thank you very much, and thanks to the organisers. So today I'm going to tell you a little bit about the work in our lab looking at the eardrum, which I hope I'll be able to convince you is a really fascinating structure. And so when we're looking at the eardrum, we've mostly looking at mouse, but I'm going to tell you a little bit about our human work as well. And we're talking about this structure over here in the ear. Let me just check you can see my pointer. Great. So pretty much all land animals have what's called a tympanic ear, which is an air-filled middle ear. And we've got an eardrum on one side where sound comes in, and the eardrum vibrates, and that vibration is picked up by a little middle ear bone, which then transfers that information over to the inner ear. And if you are a bird or a reptile, you've just got one little bone in your ear. If you're a mammal, you've got three little bones. But actually, the eardrum itself also appears to be very different. And it's now believed that we've got independent formation of eardrums and air-filled middle ears in land vertebrates. And that has a sort of interesting knock-on effect, because it means that a reptile ear didn't turn into a mammalian ear. They are distinct features. So for us, with our eardrums, very important for us to be able to hear. We need to have an intact eardrum, and they perforate relatively easily. So here's an example of a human eardrum with a big hole in it. And in most cases, those holes will heal. And usually the hole is formed because it could be a loud noise, it could be a blast injury, so pressure change, it could be a head injury or an infection such as otitis media or glue ear. So often in those cases with particularly children who have middle ear infections and have glue ear, little holes are actually intentionally made in the eardrum with the insertion of grommets to allow fluid to come out of the ear. And that's sort of possible because these holes generally do heal. And it takes about 4 weeks for the healing process to happen. However, you do get this effect of scarring. So this is an eardrum of an individual who has had obviously holes in their ears in 2 different regions, and they've healed, but we've got scar tissue. And in about 6% of cases, you don't get healing at all. And that can lead to pain, tinnitus, which is the ringing in your ear, and then current infections. And in that case, people put little sort of grafts of tissue from around the body and try and patch up the hole. That doesn't always work. And there's some recent study looking at the use of FGF, so fibroblast growth factor ear drops. But it's all very well doing things like putting FGF eardrops into somebody's ear, but really you need to know a little bit more about the biology of the eardrum. What's going on, what cells are there. The fact that those eardrum perforations heal suggests that actually the eardrum is pretty good at healing, and maybe there's a stem cell population, for example, in there ready to heal a hole. So I'm just going to talk you through a couple bits of anatomy about the ear and the eardrum, just so that you know what you're looking at when you see my pictures. So here is one of the middle ear ossicles. This is the malleus, and this is a mouse ear. And it's got a structure called the manubrium that comes out and inserts directly into the eardrum, which is here. And then we've got a bony structure called the annulus that holds the eardrum and keeps it taut. And in fact, if you look at a mammal's eardrum, and again, this is a mouse, it's divided up into 2 different regions. And those 2 different regions are very different from a morphology point of view. They're different from a sort of disease progression point of view. And they're called the pars tensor, which is this main bit over here. This is really a tight, thin part of the eardrum. And this is where sound waves are going to be transferred into the middle ear and picked up by this manubrium. But we've got this other bit over here called the pars flaccida, which is much more flexible, and that's involved in adapting to pressure changes. And when we look at an ear, I think it's interesting to note that actually there's lots of variations across the animal kingdom. So, for example, our ears look a little bit like a cat's ear. We've got a rather small pars flaccida up here and a big pars tensor. But if you're something like a sheep, over here, you've got a giant pars flaccida as well as your pars tensor. So part of the time we try and look at humans and human tissue as much as possible, because actually we really want to know about how the human ear is able to regenerate. So a little bit of sort of development. So you've got to get an eardrum, this very thin structure. And it's created as a bit of a sandwich. So we've got cells from the inside of the embryo that are endodermally derived. This is the first pharyngeal pouch. And we've got cells from the ectoderm on the outside. And these sort of push together. And we've got our mesenchyme that's neural crest derived in the middle. And these come together until in an adult form we get this sort of squashed layer. So we've got 3 layers. Ectoderm on the outside, neural crest in the middle, and then endoderm on the inside. Now, it doesn't actually happen quite like this schematic suggests. This is a human embryonic ear, and this is the ear canal coming in here. And what I want to just sort of highlight is the fact that the bit of the ear canal that's coming up and is going to form the eardrum in this region here. So this is our sandwich, ectoderm, neural crest, and endoderm. That it's a solid structure. So that schematic sort of suggested that it was open all the time, but it's a solid structure. So this needs to be open up so that we get an open ear canal, and the one layer of it will form part of the eardrum. And so here's again human ear. This is now at 17 weeks, and we're zooming in on that bit of the ear canal, and it's split open. But importantly, it's split open very asymmetrically. So it's left behind a very thin layer that's going to form the eardrum on this side. And we can see that in a little bit more detail here. So the same age, 17 weeks, we've got the ear canal is split open. Here's our eardrum now forming. And the eardrum, if we zoom in on it, we can see that it's got this very thin layers of ectoderm shown in red and green. And then we've got our middle layer, and we've got blood vessels sitting in our middle layer. And then we have a very thin inner layer made out of endoderm. Okay, so we've made an eardrum. If you look at where potential stem cell populations might be in the eardrum, the first point of call is to look at things like label retaining cells. And so you can do that with your BRDU pulse chase experiments. And when you follow and after the chase, you see that there's 2 regions where you see label retaining cells. So it suggests a stem cell niche. And that's in the middle of the eardrum, where this bony manubrium attaches to the eardrum, and on the edges, so here and here. That's an area also where there's really rich vasculature. So this is a mouse eardrum, really lovely imaging showing all the blood vessel network. And we can see it's very strong all the way around the eardrum, doesn't really penetrate within the eardrum. It's along this side over here and then down the middle. And so one of the things we can start looking at is to say, well, if there is a potential stem cell, group of stem cells in the ear, do they express typical markers that we might expect to find in a stem cell niche. And so you can look at lots of different systems, for example, the gut, and say, well, what signaling pathways are really essential for many stem cell niches, and one of those is the Wnt pathway. And so this is a readout of Wnt signaling using AXIN2, and we see that we've got a really similar expression pattern to those label retaining cells. So we've got expression in this region, which is shown this bit here around the manubrium, and this part over here around the outside of the drum. So it looks like Wnt's might be involved in this potential stem cell niche. You can then also look at other markers. So this is looking at two of the SOX genes. We've got SOX9 and SOX2. And they've got very interesting expression patterns. They're quite distinct from each other. SOX9 here is expressed around the manubrium, along this part of the border between the pars tensor and the pars flaccida, so it should be PF, all the way around. So again, very similar to the AXIN2 expression and overlapping with those label retaining cells and the vasculature. In contrast, SOX2 is actually expressed within the eardrum and very widely throughout the middle ear. And actually, it looks like SOX2, at least in the middle ear epithelium, is not necessarily associated with being a stem cell population because all those SOX2 positive cells are differentiated ciliated cells. So a bit like in the inner ear, where your SOX2 population actually labels a differentiated cell, which is the supporting cell, it's possible that these are, call them, stem cells with a day job where they might be able to de-differentiate in times of need. One thing that's very different about SOX2 and SOX9 is the fact that we've got loads and loads of SOX2 in this pars flaccida, where we don't have any SOX9 at all. So one of the things we've started to do, we concentrated on SOX2 because of its association in many other systems, and we looked at if we lineage traced the SOX2, what was it making? So this is just showing us scattered SOX2 cells 5 days after we've got recombination with an injection of tamoxifen. And after a month, we see we've got many more cells. In fact, almost the whole of the inner layer of the eardrum is labeled with these SOX2 cells. And interesting, it is just this thin inner layer with these very large shaped cells that are pulled out and form this endodermal mucosal layer. But then when we leave those mice for up to 10 months, we see actually we've really lost the signal. So that would indicate that actually the SOX2 cells are not, during normal homeostasis, are not acting as a stem cell population. Perhaps they're more like a progenitor cell population that is able to create much of the eardrum tissue, but doesn't have that stem cell properties. And we've just started doing lots of rather fun imaging using confetti mice and looking at this sort of question of where the stem cell niches are. This gives you a great opportunity to follow lots of different populations of cells and then image how those clones grow and interact and move during the process of eardrum homeostasis. So I'm now going to move on to eardrum repair. So one of the things we can do is go in and make a hole in the eardrum. So here is an example hole that's been made in the pars tensor. And in the mouse, holes heal really, really well. It's almost impossible to make a hole that doesn't heal in a mouse. So they have really good regenerative repair tendencies in this region, but they do form scar tissue. So after 10 days, this is where the hole would have been, and we can see it's obvious where we made a perforation. And one of the things we've been trying in the lab is to take these eardrums out and actually put them in a little dish, and then be able to watch the repair process sitting in a dish, which obviously is really useful to be able to track cells and follow that repair. So when you make a hole, there is an almost immediate reaction from the surrounding tissue. This is looking at proliferation rates, and this is the work of Aaron Thward's lab at UCSF. And I think it just really beautifully shows that when you make a hole, you suddenly have this huge upregulation of proliferation by all these green cells, and you get a lot that's all around the outside of the eardrum and in the center. So these are the regions where we saw those actin-2 positive cells and SOX9 positive cells. So this is the region that's being targeted and starts upregulating and proliferating. If we look at the vasculature, we see that we get a ingrowth of vasculature, which is quite usual in a lot of wound repair situations. And so by 10 days, even though now we actually can't see where the proliferation was because the repair has happened, we see all these blood vessels still there, and then they start to retract back to lining the outside of the ring. Macrophages are also involved, and one of the things I think is really interesting is that there are huge numbers of resident macrophages that just sit in our eardrums. And the places where you find tissues where you find this sort of resident macrophages are often tissues that have preparing for sort of onslaught and are often damaged. And I think the fact that we see these resident macrophages just shows us that this is a tissue that actually has to repair itself fairly often. And when we make a hole, we get a depletion of macrophages in the surrounding area as they sort of zoom in on the hole, the perforation site. And I think it's also important to really understand what cells are responding when you cause an injury. And this is some pictures again from Aaron Thward's lab, which I think just rather nicely show that this is where a hole is being made. And this is a few days, 3 days after a hole has been made. And we can see this very thin group of cells in both of these samples that have started to sort of migrate in and try and bridge the gap. And you could imagine it's quite difficult when you've made a hole in a structure like an eardrum that's under a lot of tension, that actually when you first make a hole, the hole grows because everything sort of recoils. And then you have this, as I say, very thin layer of cells that starts moving in and tries to bridge the gap. So we've been interested in what those cells are, and are they the ectoderm cells on the outside? Are they the endoderm cells on the inside? Is it the neural crest cells in the middle that start getting in on the action? And so to look at that, we've been going through various of our reporter mice to be able to follow the wound repair process. So here's some examples just looking at a label for the middle layer. So this is a neural crest marking mice. And in this case, this is the hole, and it's 5 days after the hole has been made and it started to repair. And so all these cells here have moved in and started to repair this hole. And what I want to point out is that these red cells on the outside are actually quite far away from this, this wound site. You can see that clearer here, that they're far away. And so the neural crest cells are certainly not the first to respond to the damage. And we now think it's the ectodermal cells on the outside that are the ones that are stimulated to start moving in quickly, filling the gap, removing the hole. And then at a later stage, the neural crest cells move in, and we can see here those neural crest cells that have sort of accumulated at the wound site. So now we don't have a hole anymore, but we've got a large number of neural crest cells that are all sitting at the wound site. And we think that this probably contributes to this scar formation that we can see, for example, over in this ear. So what we'd now like to do is take these kind of models and start looking at potentially how some of those stem cells might be reacting to formation of a wound. So, for example, if we could repeat these kind of experiments, we could do it in our Wnt-signaling reporter mice. We can do it in lineage tracing mice for our SOX2 and our SOX9 mice, and see what the contribution of those cells is. Is it an early contribution? Are these cells coming on like the neural crest a little bit later in the day? And then it also gives us the opportunity to then start manipulating the system. So we have, for example, for the Wnt pathway, lots of ability to delete Wnt signaling, enhance Wnt signaling, and look at how it affects wound closure. So I'll finish there, but hopefully I've been able to show you that during development, the eardrum has a fairly complex development. And one of the really key things is that we have to have this asymmetrical opening of the ear canal so that we only leave a really thin layer of ectoderm on the outside of the ear canal. We're proposing that there is a population of stem cells, that there is a stem cell niche in the eardrum, and that is around the annulus, around the eardrum, and in the center of the manubrium. And that's where we see the labral retaining cells. It's where we see the SOX9 cells and the axin 2 cells. When we've lineage labeled the SOX2 population, we don't think they are actually a stem cell population because they are depleted over time. When you make a wound, there's a different effect with the different layers. So the ectoderm layer seems to really activate and move in first. And we'd like to look at how we can help sort of stimulate repair of the eardrum, so that potentially there are new treatments for those holes that don't heal in human patients. So thank you very much for listening. And this is my group, and I particularly want to thank Juan over here and Mona and Olivia, who's just started in the lab. Thanks very much.
Great. Thank you, Abigail. Thanks for also stepping in with such a short notice. We're running a bit late, as we all know, because of this technical glitch. So there are just 2 quick questions. I might start with one from the chat. I think it's probably from Rob. And the question is, what is the turnover of the epithelium during normal homeostasis relative to other organs? He says that if the lineage tracing studies suggest turnover is happening, but I wonder how quickly.
Yeah, it's a really good question. And actually, I think one of the things that is clear is that the different layers of the eardrum turn over in a completely different way. So they're not in a sort of uniform timetable. And on the outside, so the ectoderm layer, they're turning over probably once a month.
Okay.
And all the cells, and you can think about your ear as a bit of a sort of cul-de-sac. So all the cells, they're coming from the eardrum, they're proliferating, and then they're all being sort of shed out of your ear at the end of the day.
Great, thank you. And then there is another question from Nikki Kuma. I'm not sure I understand this question completely, but I think the question is, when the drum hole tries to heal, isn't there a danger of developing autoimmune disease? is the ear isolated from the immune system like the eye area? I guess, in other words, probably they're asking if the ear and the eardrum is an immune privileged site.
Yeah, it's. I don't think it is, but it's certainly an understudied area. So it would be something we'd like to have a look. And the immune system is really important. I mean, one of the reasons why eardrums don't heal is in patients that have compromised immune systems.
Great. Thank you again.